Hi everyone, this is Satish and today we are going to discuss about measurement of plant diseases. Before we go into our topic, we have to know what is plant diseases. Disease is a malfunctioning of a host cells caused due to continuous irritation by a pathogen which results in the expression of symptom. So symptom in plants are expressed in two terms. One is localized symptom, another one is systemic symptom. In case of localized symptom, if a symptom is confined to a particular part of the plant, then it is known as localized symptom. Examples are leaf spots. In case of systemic symptom, if the infection which can be translocated from the point of infection to the other parts of the plant, means it can move from one place to another place, then it is known as systemic diseases or systemic symptom. Example is loosening of wheat. So while coming to our topic, why we need to measure disease? We need to measure disease to know the prevalence and extent of damage caused by a disease, to develop effective measurement strategies like integrated disease management. So disease is measured in terms of intensity. So disease intensity can be expressed either as two models. There are two models to measure intensity. First model is disease incidence and second model is disease severity. So selection of or choosing of either one of these models depends upon the type of the disease and our objective study for the research purpose. So first one is disease incidence. So disease incidence can be expressed as number of infected plants by total number of accessed plants into 100. So when we have to go for disease incidence calculation is when whole plant is affected. When the whole plant is affected, like systemic diseases are there. Examples are systemic diseases such as viruses, wilts, smuts, fruit rots. When the whole plant is affected, in that cases we need to go for calculating disease incidence. That is, disease incidence is equal to number of disease plants by total number of plants accessed into 100. Next method is to measure severity. So disease severity is equal to sum of all disease rating by total number of rating into maximum disease grade into 100. So this disease severity can be expressed in two types. One is by standard area diagrams and another one is through descriptive case. In case of standard area diagrams, there will be a standard pictorial diagrams. By comparing these diagrams, we need to access the leaf that is affected from the field. We need to compare with the standard diagrams and we need to give them percentage. For example, the pictures given here is on the A, it is given like leaf rust of cereals. On the B, it is stem rust of cereals. In case of A, that is having leaf rust of cereals, first diagram which is showing percent infection is 1%, second leaf which is having 5% infection, third leaf which is having 15% infection, and fourth leaf which is having 25% infection. By visualizing the standard pictorial diagrams, we need to compare the infected leaves that is bought from the field and we need to give them percentage, right? And these are the standard pictorial diagrams for septoria, gloom blotch of wheat and late blight of potato. So infection percentage 10%, 25%, 50% is given for septoria, gloom blotch of wheat, and late blight of potato we have 1%, 10%, 25% infection and 50% infection. By seeing these pictorial pictures, and we have to compare the infected leaves that are bought from the field and we need to give them this percentage. Next method is descriptive scales. So standard descriptive scales are given for measurement of this disease. For example, we need to give them as a grades. So standard scales are given from grade from 0 to 9. So 0 grade, 1 grade, 3 grade, 5 grade, 7 grade, 9 grade is given. So 0 grade, the percent infection on the leaf and percent infection in pod. If it is 0 grade, then no disease is there. If 1 grade, less than 1% infection on the leaf. In case of pod, that is pinpoint pods on the pod. If it is third grade, then percent infection on the leaf is 1 to 10%, the percent infection on the leaf is less than 1%. If the grade is 5, then the percent infection on the leaf is 11 to 25% infection, and percent infection on the pod is less than 1%. If the grade is 5, percent infection is 11 to 25%, on the pod is 1 to 10%. So if the grade is 9, so 9 is our maximum grade. So on the leaf the infection is greater than or more than 51%, and on the pod is greater than or more than 26% infection. So here our grade, our scale, descriptive scale, the maximum grade is 9, which is having more than 50% infection on leaf and more than 25% infection on the pod. So in this picture we can see anthracnose disease of bean, which is allotted, grades are allotted based on the percent infection on the pod. So on the first picture, on the first pod, 0 grade is given because there is no disease on the pod. On the second one, 1 grade is given because we have small pinheaded spots. On the third grade, so on the third grade, the infection on the pod is less than 1%. On the fifth grade, the percent infection is between 1 to 10%. On the 7th grade, the infection on the pod is 11 to 25%. On the 9th grade, the infection is more than 25%. So based on the percentage infection, the grades are allotted to these pods. So once we have collected all the data, we need to allot those data, or we need to substitute those data into the severity formula. So we all know that disease severity is equal to sum of rating by total number of rating into maximum grade into 100. So first we know all grades, disease grades from 0 to 9, that is 0, 1, 3, 5, 7 and 9. And next one is total rating. Total rating means we need to know how many pods we have seen at 0 grade. So first we have to compare that. On 0 grade we have observed 5 pods, right? So 5 pods are having no disease, and 1 grade means on 1 grade we have seen small pinheads spots like 5 we have observed. On the third grade, less than 1% infection on the pods, number of pods that we have observed is 8. So on the 5th grade, number of pods we have observed is 4. On the 7th grade, number of pods means this total number is not constant. It is depends upon the total pods or total leaves that are being affected. So the total number is not constant. It depends on the field number. So on the ninth grade, which is maximum grade, so 4 pods are being observed which is having more than 26%. So this total rating is not constant. It depends upon the infection in the field area. So here one example is given like that only, but this total rating is not constant. When it comes to number of rating, so number of rating is equal to disease grade into total rating. So first one on the 0 grade, total plants that are total pods that are observed are 5. So 0 into 5 is 0. On the second one, on the 1 grade we have observed 5 pods. So 1 into 5, 5. On the third grade, 8 pods we have observed. Then 3 into 8, 24. On the fifth one, 5 into 4, 20. On the next one, 7 into 8, 56. Next, last grade, maximum grade, 9 into 4, 36. So we have to plus all these grades like 0 plus 5 plus 24 plus 20 plus 56 plus 36 is equal to 186 is a sum of all ratings. So total rating also we need to add all the ratings, that is 5 plus 5 plus 8 plus 4 plus 8 plus 4, that gives you 44. So total rating is 34 and sum of all rating is 186, and we have maximum grade, that is 9. So 9 is a maximum grade. So substitute these all values into the formula. Disease severity is equal to sum of all rating, that is 186, by total rating, that is 34, into maximum grade, we know that 9, into 100. So Sum of all disease rating by total rating into maximum grade into 100, that gives you 186 by 34 into 9 into 100, that gives you 60% disease severity. So thank you guys, thank you for watching.